Hi, my name is Adarsh Mambadi, and I'm a junior at Archbishop Midi High School.
My project is called Predicting Regional Mutation Rates of COVID-19 using Machine Learning and Designing siRNA Therapeutics using Phylogenetics. Demographic, environmental, and SARS-CoV-2 mutation data for all 50 states were compiled. If a factor had very little variance, they were removed from the dataset and from further analysis. Here you can see the code that does this. Then t-tests using a p-value adjusted using Bonferroni corrections identified individual features that are significantly associated with mutation rates. Here's the code that does this step. For each of the 137 features that were identified in the previous step, linear regression models were run in order to quantify this significant relationship. Here's the code that completes this step, and here is a sample example of one linear regression model, and again, here is a sample of all the notable factors and their coefficients. While Stage 1 only identified individual features that have a significant relationship with COVID-19 mutations, Stage 2 identifies a predictive model that takes all these individual features and identifies the relationship between them and COVID-19 mutations. As you can see here, the best model was elastic net regression, which has an accuracy of 87%. In the two performance curves, we see that the model had an extremely high area under the curve, which meant that the model is both able to distinguish between states that have a high and low mutation rate and made very few predictive errors. In Figure 3, we see the test that the model took in which it got 13 out of 15 correct, and in Figure 4, we can see the 10 features that the model used the most in order to determine whether a state had a high or low mutation rate. Lastly, 16 region-specific siRNA molecules were developed to demonstrate the model's ability to help create localized treatments. In this three-pronged approach, scientists will be able to take preemptive measures and prevent outbreaks caused by pathogenic mutations, just like the current COVID-19 variants. Thank you for listening.